Hi, good afternoon. This is Bharat Pathinji. Uh, sure, I know most of you, but we'll go through the introduction in a few minutes since we only have 14 people. Okay, the title of the talk is Human Evolution. So, and this is a three-session class in which we will try to understand what is new in human evolution that has taken place since the days when we went to high school and studied all about paleontology and other excavations. Okay, now let me focus this properly so I can show this. Now, this basic outline of the classes will talk about primary difference between our nearest cousins, chimpanzees. Then we'll briefly talk about various hominoids, primarily focusing on Neanderthals and the new species, Denisovians, and a couple of other new ones which have been discovered in South Africa and the Philippines and the Indonesian islands recently. And then we'll talk about some of the effects of genes have on evolution of human body, which is far from perfect mechanism. And then we'll talk about the health of genes, evolution on health and disease. Okay, so hopefully we'll be able to go through a lot of this material in time. Okay, now this is an interesting book about the origin of human beings by an English woman who is primarily an anatomist, and may remind you of Incredible Likeness of Being. There was a movie starring Daniel Day-Lewis a long time ago, but that's digressing. Okay, so now just to show that human beings have always been interested in evolution, and in the early days, obviously, humans created God in his own image and then gave God the credit for creating man in his image. Okay, and this is the classic reading from Genesis, which all of you are very familiar with. But not to be outdone, the man who was the dawn of English literature, this is the famous quote from Hamlet: "What a piece of work is man, how noble in his reason, how infinite in faculties," and so on and so forth. You can read about it. But then eventually, this is science, after all, a science class. And when we talk about evolution, it would be heresy if we didn't start with Darwin. And this is Darwin's quote. So whether we like it or not, depending upon how religious or non-religious you are, after all, we start out as animal. And if you go further and further back from the beginning of Earth, we started out as a single cell organism. Okay, or in our case, probably two cells which joined together from your mom and dad and made you what is you. Okay, so here again, I'll have to keep moving my slide because, okay, this was focusing is not correct on this thing. So I will move to make sure you get the full view. Now, this was the classic concept going back to the Greeks in the Aristotelian days as to how he had given a very logical thought as to how things evolved from nothing to minerals to plants and gradually going up to man being on the highest amongst the realm of becoming in the realm of being. So they obviously, the concept of demons and angels was very much in vogue. And so he gave enough credit, and that may have sadly. And this is again another quote from a well-known English author: "A hen is only an egg's way of making another egg." So that's so that just fits in nicely with the recent concept of Richard Dawkins about evolution being based under a selfish gene, and the gene has only interest in reproducing and does not care for as to what exactly happens to you. And as long as, so the main interest of one life is to make another life. Okay, and but again, so this has all should be seen in the perspective of evolution. This was a famous quote by a Russian biologist. And more recently, we've become more cynical. So this is our current view as to go back. We effed up everything, and which is probably not too far from the truth, looking at the current state of the world and the dominant species who is on its zealously trying to ruin Gaia, or and the concept that has been proposed in last 30, 40 years is that the Earth is a living, breathing organisms, and we are all interconnected, starting from the smallest cells in bacteria that live in your stomach to the fungi that live at the bottom of the trees to the oceans and so on and so forth. And it does make a lot of sense. Going back to Darwin, whose phenomenal work was The Origin of Species, after his only a 5-week journey on the HMS Beagle around the coast of South America, and after seeing the various birds, he came up with the concept, and for whatever reasons, he chose to sit on it for a long time. And finally, when the Royal Society got a similar article, article to be paper to be published from a hardworking biologist in the Dutch Indonesia named Arthur Wallace, that his Darwin's friends, the Royal Society, published. The difference between Darwin and Wallace was Darwin was independently wealthy, being heir apparent of the Wedgwood family, and his father was a respected and a wealthy doctor as well as a poet. Okay, so then he published his magnum opus, The Origin of Species, only later to be followed by Descent of Man, in which he answered all the concerns that the other people had about his theory as to the so-called missing link. And prior to that, you may have studied that in school that Jean-Baptiste Lamarck had proposed the ideas of evolution as to how the evolution responds to environment. And he wasn't exactly trying to propose, but in high school biology, it became that Lamarck was wrong and Darwin was right. So that was not actually quite the case. And in fact, as we'll talk about later with the new advent of science of epigenetics, Lamarck may be more correct than he was given credit for. Okay, so Darwin finally clarified that this descent of characters does take place with imperfect modifications. Okay, and so this was relatively, a new concept that he finally answered in the 6th edition of his books when he was questioned by a self-taught lawyer come naturalist, as they used to call themselves, and about the explanation as to how many. Okay, so this is, so, and obviously they had to answer question about the various odd things that they saw or observed in nature, such as peacock's tail, as well as frogs with low-pitched sounds, and and the trade-off that occurs between getting eaten by other animals versus catching more flies. And so there are varieties of trade-offs that takes place, and the similar trade-off that some of you may recall from my class on aging, that we talked about, that some of the price that we pay for aging is such as a cancer type of thing. And also there are varieties of trade-off that evolution has said, called, and the fancy word that we may recall was called an isotropic pleiotrophy type of thing. So that was the word that was published in the other literature. So, and so this is going back to, because I think I might have missed a point. So, okay, so it just goes to show that the evolution of you and I took place from a single cell to, and we're still evolving. And it is apparent by the fact that even after age 70, most of us are trying to attend class and evolve further and form new synapses. Okay, so this is, and so this is just important that not knowing evolution for physicians is like doing engineering without knowing physics. Okay, so that was a fun statement by one of the evolutionary medicine, which is a relatively new branch that has popped up in recent past. And I got a new opportunity almost to learn a lot about it within the last few weeks, especially after we sent out the survey as to what the class was more interested in, as to whether primarily evolution or genes as effects to health. Okay, so, and now Darwin was puzzled by the problem with inheritance as to how they, obviously, since the times immemorial, men were given the credit for reproduction and women were just considered as empty vessel who provide the fertile soil for breeding. And it was the men who determined all the heredity. And obviously, and it took until early 1600 when the ovum was visualized with available microscopy in those days that the role of women became clear. And Darwin proposed some mechanics of inheritance, and he coined a word called gametes to explain inheritance. But in those days, he did not have access to internet, so he was unable to, or was not aware of the paper that was published by a frustrated teacher or a failed teacher, a monk in Czechoslovakia, in an abbey, who did research on pea plants and methodically bred the very characteristics of the plant from tall, short to wrinkled versus non-wrinkled and based on that, established the primary laws of heredity and determining that the inheritance of tall characteristics was more dominant, as explained by in a simple terms that if you took tall and bred with short, the tall will dominate, and if they have the same number of progeny, then the ratio would be 3:1, so to speak. So that would be the dominant ratio that was established, and that is something called nowadays called classical genetics. And in those, obviously, they had not visualized the gene, and this was way before DNA isolation. Now, the Lamarckian principle, and unfortunately, his work was lost for about 30, 40 years, and was rediscovered or reproduced independently by some German botanists. And finally, the idea of gene is accepted in Western science. And a naturalist, zoologist, philosopher named Bateson coined the term genetics for the first time in around 1875 or even 1890. I forget the exact time. Okay, so this is, explains. And now from gene onwards, the concept has evolved further in more recent time. And the Darwinian evolution has been replaced by various other concepts of something called neo-Darwinism. And some of you may have read the books by Stephen Jay Gould, who explained about something called the punctated equilibrium and a rapid burst that takes place. And primarily, he did work on excavation called Burgess Shell, where they saw an explosion of new species appear in a particular layer of geological digs. And so there are varieties of new concepts, and there are other pioneering geneticists who have advanced the thinking. And most recently, all of us grew up in the 50s with the invention of computers. The idea of information being paramount is being incorporated as to finally the life is compared with, because every generation has compared life and everything with the technology available at that time. As initially it was compared with steam engine versus looms, and recently we are comparing as life is primarily digital. But if you get into some serious thinking, it's maybe a combination of digital plus analog, and we'll talk more about it later on as time permits. Okay, so, and luckily for the origin of human species, on one hand, we are a lucky accident due to the fact that only a relatively small number of people as few as 1,000, or maybe by the biggest estimate of about 10,000 migrated out of Africa and managed to populate the whole world in a few thousand years, traveling all the way to Australia and Tasmania after about 60,000 years. And in the last 200 years have exploded from 1 billion to as much as 7 billion, and on a way to become 9 billion. At the same time, dominating and wiping out a lot of other species. So this is, but that again, the other major accident that facilitated the rise of mammals was asteroid that struck the peninsula in the Yucatan in the ocean near the Yucatan Peninsula, and wiped out the dinosaurs, and that has been well proven. And the discovery of that is itself another interesting story. Okay, so any questions? I will stop for a few seconds. If not, we'll continue. Okay, and obviously, in terms of evolution, we started out as the last common ancestor, which hasn't been isolated, and the earliest evidence of life are the various mats or stromatolites that are discovered in Australia, which are algae that grow on the ocean front. Okay, and there's still, for those of you who are interested in the origin of life, you have to wait till I offer my other class sometime in spring of this year. And again, the evolutionary scientists have wisely avoided the question of answering how do viruses fit in, because if you ask several biologists, they will hedge their bet as to viruses are alive or not, either yes or no. So the jury on that is still out. And that has become a very relevant question in view of the recent pandemic. Okay, and now this was the classic evolution that was proposed by Carl Linnaeus even several hundred years before. He was a great classifier of all the biological species, and obviously must have more than his share of obsessive compulsive element. And this was his classification, starting from kingdom to phylum to class, order, family, genus, and species. And obviously, we are the Homo sapiens species, and a genus Homo, and we're a part of the hominoid family. And then we are primates, obviously, and we're mammals. And obviously, we are chordate, means we have a spinal cord of some sort. And last but not the least, we are not all plants. Okay, so, and this is a mnemonic that was taught in the early days of biology, that dear King Philip came over for a good soup. Okay, so that's kind of rather silly way of describing it, but there you have it. So, so we explained to you about the classification of humans in the classical way. And now this was the overview of the tree, as this is probably maybe hard to see. And I'll see if I can, without messing it up, I can magnify it. Okay, let's see if I can bring my magnifying glass. Yeah, oh yeah, it works. Pedigree. So again, as in the Aristotelian concept of man being supreme as at the top, and the other families are at the bottom. Okay, but this was not all as cut and dry as this is the more modern concept of evolutionary tree, because as you can see that this was after the advent of microscope. The bacteria and take up a large amount of space, and this is our cutoff part of the things, which includes winds down all the way to bacteria and even more primitive organisms called archaea, that they managed to survive in extreme hot weather, even at the bottom of the Yellowstone geysers. And then the evolution occurs with primarily boneless fish, and then various sharks, fish, and then again moving on to amphibians, reptiles, birds, after the demise of dinosaurs, and then mammals. And we're somewhere at the last end of, somewhere at the tail end of this thing. So realizing that life appeared very shortly after the origin of Earth, as many as 305 billion years ago. We have very little evidence of history. We only know about last 0.5 billion years of history after the Cambrian explosion somewhere here, and then the human beings, that is current us, Homo sapiens, appeared within the only last about a couple of 100,000 years, give or take a few. So we are relatively new species, and how long we will last, that time will tell. Okay, and we talked about the crater, and this is a view of the classical evolutionary or paleontology. And these are some of the supercontinent Pangaea that formed about 230 million years ago, with the rise of plants, reptiles, and the ferns that dominated the Earth. And then the other marine life got wiped out with the explosion and the mass extinction that took place. And as much as 90% of marine life was wiped out. And so it was a lucky survival. Dinosaurs and mammals gradually evolved, but that time the mammals were mostly nocturnal and lived, and they were small size, and the dinosaurs dominated the Earth from almost 200 million years ago to as recently as 60,000 years ago. Okay, and so this was the state of affairs. And then again, continuing as the continental drift continued in the present time, the flowers appear rather late, and the dinosaurs continued to dominate. The fish evolved further and diversified. Okay, and then finally the big asteroid strike that we talked about, which was a lucky break for us, bad for them. Okay, and in Africa, the gradual evolution from various apes took place, and to the well-known hominoid, Lucy, and even prior to that, another species was identified, which started us on the human journey, and the varieties of modifications that took place, which got us into present state. So, okay, so the varieties of fossils that have been isolated are as far back as 233 million, thousand years ago. And this was published as recently as January 12 in the Nature magazine. Okay, and so the evolution, now the interesting question is, has evolution actually stopped? Are we still evolving? Okay, and the answer is it has slowed down perhaps, but yes, it is occurring. The people who do the work on it have said that even in human beings, we are evolving. How is that evidence? Because the average body temperature is lower than it what used to be. And this may be primarily due to less inflammation, because we are clean on the outside and inside, and we live in a sterile, hygienic world, so to speak, after the Industrial Revolution. And less inflammation means there is less chance of bodies fever reaction. At the same time, other good example is that lactose tolerance developed spontaneously in 3 separate areas: Northern Europe, in the Mesopotamian era, and in Africa, because the Maasai, even though they never left Africa, they are still able to use dairy products. And at the same time, human bones have become lighter. And so the varieties of changes are occurring. When it comes to other species of animals, the evidence is very, very obvious. Just to give you an example, a small bird called cliff swifts. Okay, they in the urban population, when they landed to catch on the road, they were being overrun by cars, after the invention of automobiles. So they've gradually, their wings have shortened so that their takeoff area is relatively small. And that was surprising to me. At the same time, even other animals have evolved. With the rise in poaching in African elephants, their tusk size have decreased. And not to be outdone, even the smallest insects have evolved, such as there is actually, believe it or not, there's a London underground mosquito, and that just lives underground, never comes to the surface, and survives on biting mice, basically. And it has been given a special name called Culex mollustus. Okay, so there are varieties of evidence that's still taking place of evolution, just to get the point across. Okay, yes, and this is important to say that the last recent hominid human remains have been dated as far back as, and the dating has become more and more precise. Okay, and in terms of various origin, people in the ancient before the Enlightenment was all had given several thoughts that the human sperm carried a tiny little uh, all the all the information needed for formation of a baby. And because obviously, as I explained earlier, that the women were not given any credit for contributing to the next generation. But that has been disproven solidly with the invention of modern science, that with the invention of microscope, we became aware of the presence of cells, and which formed what composite cells go on to form tissues. And on the other side, cells contain nucleus, which has the genetic code. So this is, the circle that goes on between the genes that carry the information, forming the cells, and the complex cells go on to form tissues, and tissues go on to form human beings. Okay, so this is the more accepted concept of epigenesis, or formation of new life, that is currently accepted without doubt. And again, the evolution and extinction takes place. And this nice little chart that may sound weak. Nowadays, the size of the graph explains the number of species that were there, and starting from the Paleozoic area to Mesozoic to Cenozoic area, and that is going back to 250 million years ago, as to varieties of boneless fish appeared, and then gradually the more complex animals evolved, and finally the mammalian species, which there are about 5,000 or so mammalian species. Okay, they're around, and most of them, the normal rule is that a lot of these species have gone extinct. So very few actually have managed to survive until the present time. So that does not speak very well for us human beings as to how long we will be around. That time will tell. At the same time, these are the various extinctions that have taken place. As you can see, the extinction that took place, there have been Ordovician, Devonian, all this. The English were fascinated with geology. They did not want to mess with chemistry because they didn't want to get their hands dirty. That was left to the industrial Germans later on. And geology was rather popular. And in fact, the Royal Geological Society came to existence a lot earlier than Royal Chemical Society. So that was interesting. So that's why a lot of these geological names are English names of Devonian and Cambrian. So you can see that these are, and the Jurassic was exception due to the finding of the dinosaur fossa in the Jura Mountains of Europe. Okay, so that gives you a broader perspective on the evolution that took place. But again, we are going to concentrate here on primarily talking about chimps and humans, and the role that genes have played. Okay, now a very well-known evolutionary biologist from Harvard who's written some very impressive books for lay people, and he explains the human evolution, to summarize as briefly as one could, in various 5 major transformations that took place. The first one being bipedalism, that's walking on 2 feet, living in open habitats instead of forests and jungles, living in the open savanna, then gradually developing hunting and gathering, in working in groups, and then that led to the rise of sharing food and more energy being available, and that finally, with the advent of cooking, gave rise to better brains. So these are the major transformations that led to forming of us. And so we are originally bipeds, and the earliest were Australopithecus, which was about somewhere around 3 million years ago, and then gradual cooperation rose to the rise of Homo species homo that took place. And then we will talk more about Neanderthal. So evolution, as we know it, has two main characteristics. The first is random changes that take place in your genome, okay, which is a rather blind and totally random, as we call it. The other is very smart and selective. That is, even if there is a slightest advantage, that particular change will be maintained and will be exacerbated. And on the other hand, nature will try to eliminate the bad gene unless there's an advantage for survival. Nature lives in the present. So, for example, it doesn't cause the same gene that may give you stronger bodies and more testosterone. Okay, that is primarily the cholesterol gene, may come to harm you as you get older, and after you have had enough kids, it may come back. And similarly, the genetic inheritance may occur either in combination or it may occur in a dominant fashion. So, okay, so these are the various ways. For example, the gene for height is a dominant gene, and it's almost 80% influence of genetics. If your dad is tall or if your mom is tall, you're going to be tall, and so on and so forth. On the other hand, several other genes for more common diseases are influenced by hundreds of genes working in a big combined orchestra at the same time. And we'll talk more about it on the last day. Okay, so there are varieties of changes that have taken place, including, and now the human evolution still continues even after birth. And that is well evidenced by the wiring of the brain that takes place from a newborn to a 3-year-old to up to 18-year-old. And even the teenage brain is still evolving. And some of the women will say that their husbands are still evolving, okay, and they're hoping that someday they'll mature. Okay, and the science of evolution is studied based on not just with naked eye anatomy or with digging up fossils and looking at geological formations, but other scientists such as chemistry with radioisotope and analyzing the crystal structure of the rocks is involved. At the same time, from the early 1900s, okay, the science of genetics comes into foray, and that has contributed immensely. And more recently, various mathematical models have been created as to after the rise of this statistics based on the population size as to how a genetic frequency will be inherited or shared has become useful for interpreting evolutionary flows. Okay. And last but not the least, varieties of the huge machine or technology for analyzing the DNA has played a role. And last and most recently, various computer-based data crunching has made a dramatic difference in comparing sequences of all sorts of species. For example, when we can compare the genomic sequences from a chicken to fish to mouse to chimpanzee to human, which would have taken scientists with, in the early days of computing, as many as 35 years, that can be done in a matter of weeks. So it's a dramatic change that has taken place. And I just thought this was a rather interesting thing, that it is not all genes and cut and dry as we may think, or as the strong people who were strong believers in so-called neo-Darwinism would like you to believe. But nature as well as nurture has both effects. The photo on the left shows an English family and the tall man who's married a tall woman, and their 2.5-year-old is looks like a 5-year-old. And at the same time, Dr. Lieberman, Dan Lieberman, whose book I mentioned, talks about the height. The Paleolithic men were relatively tall, but with the farming and infections and the less nutritious food that became available, for example, just lack of protein, because they survived mainly on cereal, and with the it changed from hunter-gatherer where they got more protein. So this average height during the rise of agriculture decreased with the industrial right and with a healthier lifestyle. it started increasing. And now it can be said that the Dutch are the tallest people in the whole of world. And at the same time, as you go close to the tropics, the heights decrease. So it's a big genetic influence. So nature and nurture are both equally important. Now, as you can see that before the rise of genetics, most of the conclusion about human evolution was based on study of growing embryos, not just human embryos, but other embryos. And the biologists, or to say be more specific, the embryologists who did the study were impressed by the similarities in early stage embryos from fish. And you can see that you don't have to be an embryologist to appreciate, but when you look at fish or salamander or tortoise, it would be hard to tell the difference if they are not well labeled. So it's not like that there is the human starts out with their own uniqueness, okay? And it just gradually over this start of confinement or pregnancy, it evolves and the fish goes on her merry way to become a fish, and humans end up being looking quite different from a salamander, okay? Now, but we also have some basic similarities, which is we are called vertebrates. When we start out with a layer of nerve tissue which goes along the back, and which was in early days called open tube, which was called a notochord, and then various gills or pharyngeal pouches appear, a tail appears, and so these similarities are well preserved across all the species of vertebrates, and then the neural tube closes in the spinal cord with the deposition of vertebra, which is the bony tissue that surrounds and protects the spinal cord, and a small area of the that still is invaginated between the vertebral bones, and that has come back to give us the pain of lumbar disc, which gets compressed, which is a small gel-like material, jelly-like material. Okay, so this is the evolution that takes place, and then the various clever embryologists, a lot of them were German, where with observation determined that there were 3 different layers of cell: ectoderm, mesoderm, and endoderm, which gave rise to different tissues. And this was considered a golden rule, that the skin and the nervous tissue form from ectoderm, your heart and liver, and gut formed from mesoderm, the inner lining of the gut formed from endoderm. And so this was considered an absolute rule of embryology. Okay, and then varieties of organs went on to develop. And this was, as you can see, that this explains what I just spoke about. The ectoderm gives rise to neural tubes and also various cells that are isolated in your adrenal glands and gives rise to the adrenal hormones, the steroids, etc. And so this is, just give you an idea about the basic embryology. And part of interestingly, the only exception being the facial tissues and the jaw muscles are formed from ectoderm. And this was proved by a woman scientist named Julia Pratt. And as for whatever reasons, and primarily the fact that she was a woman, and this went against the conventional thinking, and it required a paradigm shift that there were exceptions. In biology, there are more exceptions than rules, unlike physics. Okay, and then there are varieties of, at that time, a lot of the people were naturalists, and they collected specimens from all over the world, and some of them were totally flabbergasted or surprised to see the evolution that took place from one form to the other, and that finally convinced people. And they came across some various animals that were sent from the colonial days, and they came up with a fish that had something called lung bladder, so it managed to breathe air from the atmosphere rather than just breathing with gills. And so, and the varieties of forms that took place from the adult salamander stage from living underwater to living on land. So, and it regressed back and forth. So that's again primarily proved that evolution was a lot more complicated than they had made out to be, that it was not all straight line attempt. And this is gives you an idea about the human evolution of the hominoid species and the similarities. And now these lines that are drawn are just not drawn just willy-nilly. They are meant to explain the distance. For example, that our closest relatives are, the chimpanzees and bonobos are closely related, and we are not directly related to chimpanzee because we have not been able to locate the so-called last common ancestor. So the length of the line shows the distance between the two. And initially, the gorillas and orangutan were considered, fairly related. But again, see the distances were totally different. So some of these cladograms were initially established based on physical appearance, but with genetics we have learned different things, that the relationship that may be obvious, that may be surmised on appearance, is quite different when we look at the underlying genomic sequences. Okay, so about primates from Asia to Africa, place, and then various other apes with no tail. Or apes have strong jaws, and the gibbons and the orangutans are primarily in Southeast Asia. Gibbons are relatively smaller, okay, and they tend to be monogamous. Orangutans males are bigger, okay, and they're very similar to gorillas, okay. Bonobos are have female hierarchy. On the other hand, chimps, even though they are closely related, are a lot more aggressive, and they have multiple sexual partners, okay. And so one can say that the bonobos and the chimpanzees are like sisters, while as human beings, we are like second cousins. So from orangutans to humans, they are called hominids, so we're all apes except for gibbons, so that's how various classifications. So the last common ancestor between the two was based on molecular analysis, was about 7 million years ago. So that's when the separation took place. So this is the so-called classical anthropology or evolutionary tree. Okay, now I said it's interesting that first let's go over in details about the skeletal features between the humans and the apes. And these are very apparent, but they're worth mentioning. Primarily, the curvature of the spine is different, and this is formed by various genes that form the architecture of the various spines. And we have the three: the cervical, the thoracic, and the lumbar spine. And instead of having I'll go over jump over to the next slide to show you the picture. Okay, as you can see that the spine is single curved spine, while the humans have a nice curvature, and there's a small but definite angle of the lumbar in the lumbar region that takes place, and in the females have even a bigger curve of about 45 degrees. So you can see that the back of the women is a lot different, and that is considered as an attractive feature. And that again, the biological advances that prevents them from during pregnancy from tipping over, basically. Okay, so if it was relatively straight, then they would have a difficulty. So that just helps them maintain the balance. Okay, and now go back to the other side. The foramina magnum, which is the hole at the outside under the skull, is in a different location, and that's important to point out. That the hole and the human being is at the bottom, okay, at the center of the skull, while in the chimpanzees is on the back side. It's center C, because the head faces projecting forward. Okay, the same way the other changes are very apparent, because you can see that the pelvis shape is different. Okay, there's a lot more narrower pelvis with the almost vertical hip bone, and the angle of the femur, that's the big bone, is different as well. And obviously the hand and feet are dramatically different, as we'll talk about in a few minutes. Okay, so these are some of the dramatic changes that have taken place. And the chimps walk, when they did walk on two, they did use their knuckles and for support, and that's a motion that's called brachiate. And so they use their knuckles for support. and leaning forward. So this is, and that was useful for them to be hanging onto the trees at the same time while walking. And only in water they can perhaps walk straight with it, because there is no reason to hold on to anything, and it's a different buoyancy. Okay, and so we have a lot longer limbs, and the biggest pièce de résistance is the so-called opposable thumb, which is a dramatic difference between the chimps and us. Okay, and so these are the various evolution routine. Now, when we get down to genetics, and this is a well overcoated figure that the chimps and human genome is 98% similar, but the rest of the difference, makes us who we are. Okay, so so we are closely related to chimps, and they are related to us. But so one can say that, but the features are different, and one can say that we are sexually mature baby chimp, type of thing, and we'll see the similarities as we get down. Okay, and that is evidenced by the fact that our jaw is not sticking out like a chimp's, and we don't have something called the sagittal crest on the top. And we talk about our teeth are relatively small, and the brain sign is dramatically different. Okay, and so this is figured out because based on the changes that take place between an average generation, that every child has about 35 mutations, considering that it takes about 25 years for a next generation to appear, and the time of separation has been backward calculated to about 7 billion years ago. And the earliest hominin species that has been found in Africa is called Sahelanthropus ediba. Okay, and then afterwards came Lucy, made famous by Dr. Leakey. Okay, and these are the various cute animals. As you can see, this is just for those of you who have not been to the zoo recently. This is the gibbon, relatively small face, okay, and orangutan, and the male and female sizes are different, okay, and the gorilla, who's lives by himself generally, and there is big sexual dimorphism between the male and females, and again the chimps, and the chimps, have proven us, put us in our place, that the chimps have been well documented to use tools, and this was well demonstrated by the meticulous observation done by Jane Goodall. Okay, that all of you are, I'm sure, quite familiar. Okay, and so, and they're relatively sedentary type of thing, that they don't walk too much, and because it's hard to walk on with that type of knuckle, and the feet are not, they may be ideal for climbing trees, but they're not exactly ideally formed for walking long distances. Okay, but even though they're smaller, they're a lot stronger than us. So you don't want to arm wrestle a chimp, So compared to them, we're rather poor athletes. Okay, and so this, because of the differences that have taken place, and the earliest fossils that we talked about was, several 100,000 years ago. Okay, and some of them have been given various names, but we are not offering you a class in pure classical anthropology, but so we'll skip over that. And these are some of the skeletal features. Okay, let's go over that again. So sorry about that. Okay, yeah, here's my thing. So this is the human skeleton, and they were tall with long legs, outward pointing femur. Okay, the foramina, that's, this is the Australopithecus afarensis, male, relatively short. Okay, and you can see that the clavicle is at a somewhat different angle, and the rib cage is different, and but you can see that there are a lot more similarities than differences, and it's a dramatic departure from the chimp. Okay, that the Australopithecus have some primitive features, that the males have larger body, okay, than females, okay, and a small brain size. That's the big difference. Okay, relatively long arms. As you can see, there's like, one can see very clearly that one does not need to be an anatomist to show that there is a big, intermediate stage, and the fingers and toes are becoming longer and a little bit less curved than that of a chimpanzee. Okay, and there is the famous Lucy in between. Okay, reconstruction. Okay, Australopithecus afarensis, that she was a female, and you can see the size difference. And these are the points, various points that we talked about, Okay, and so this is a nice way to do a comparative anatomy. Okay, as to why the apes decided to get out of the trees and walk, there are various theories. Primarily, the one that were proposed with being savanna-based, because it was the climate change that took place, and the postural feeding, because it's easier, and you're able to control the body temperature. You're able to look out further the distances. So there are varieties of provisioning models that have been proposed, and they all make sense, or they can be argued back and forth from various scholars who do evolutionary medicine. Okay. And this is another slide which explains the origin of humans. And you can see that the humans evolved from the Homo habilis, which is the so-called handyman, Homo erectus, rudifrensis, that is the earlier fossils, Homo naledi, Eragaster, and then Heidelbergus, and then Neanderthal, and Denisovians, finally rising up to human beings. Okay, then getting back to the more nitty-gritty of the anatomy, that the indentation is different from a U-shaped to a nice half circle that takes place, and the changing in facial profile, that the incisors gradually became smaller, the jaw became shorter, and short jaws and highly developed pointed chin, okay, into the small jaw. Teeth were smaller because the jaw became smaller, okay. And so these are the varieties of changes that have taken place in the dentation. And so this is, this face shape change in this, okay. And this slowly, in the changes that took place came in very handy, because the larynx gradually migrated lower down, and the tongue became a lot more mobile, and the jaw became smaller. And so that gives the advantage to the rise of speech, as we got, because no matter how hard the chimp tries, they would be able to, except for making some noises, would not be able to have a nice box and magnifying the and the varieties of words and articulation that has become easier with the larynx going lower down, and the vocal cords, vibrating with the airflow at the bottom. Okay, so, and we talked about the dentation, the U shape versus parabola. Okay, and so overlap of, there is a risk with the larynx going down, because there is a big risk of choking. So one can see that the nature's design has traded one advantage at the cost of developing the risk of sudden death and choking. And these are the sciences that there are various organoids. Based on that, people are able to modify the expression of various genes in something called organoids. And there is evidence of various selected genes that can be taken place in various embryos. And this is well demonstrated in service examples. So the science has advanced quite a bit in terms of, this is a fairly recent paper that I just came across, as to how based on the amount of expression of only one or two genes in something called a signaling pathway that is associated with formation of the face. And when it is downregulated, the facial features become less prominent, and vice versa. Okay, so that just gets into a little bit more details than you would care to know. And again, this gives you the example of the finger that the chimp is, and the ape hands are a lot more suitable for grasping on to the tree branch, and the sharp claws. On the other hand, we have the opposable thumb, okay, which gives us the flexibility, to do fine motor skills. Okay, so, and the thumb size gradually became reduced, because as you can see, okay, when we're able to walk on their hand type of thing, so the thumb size kept changing type thing, and they're using fingers more as hooks. and the humans, we have the bigger thumb, okay, and which is so-called opposable thumb, which is the big dramatic. Came across, it can touch the tip of each finger, and so that gives us the rise of and then various muscles that have developed, just for control of fine motor skills of the hand. So it's a name. The muscle is called extensor pollicis brevis. It's the flex and the various muscles that work together to manipulate tools. Okay. In the same way, there's a modification that in the skeleton that is shown very, very nicely here between the chimpanzee and humans. Okay, we talked a little bit earlier about the pelvis. It's shaped as to funnel shape versus barrel shape. Okay, tapping, and the scapula is close together. Okay, and so the varieties of changes that have taken place that have made us who we are. And the foot is the developing arch that helps us support the weight. The spine we already talked about, okay, and the femur we talked about. At the same time, even the various soft tissues have development, okay, things up. And the human gut has become smaller because we eat more processed foods at the same time. And we have a better digestive enzymes because of duplication of genes. We have copies of a gene called amylase gene, which helps us digest starch in our mouth before it even reaches stomach. And various genitalia were different as well. For example, this may shock you that a gorilla, even though as big as he is, his penis is rather tiny, about only 4 centimeters, while on the other hand, chimp is smaller than humans, but the chimp testicles are about 4 times bigger than humans. So the evolution works in a mysterious ways in terms of formation of some organs. Okay, so we talked about that. And as I alluded earlier, that the shape of the baby chimp is very similar to that of a human. As you can see, there was a earlier biologist, a mathematician named R.C. Wentworth Thomas, was lived from 1860 to 1948. Okay, and he had created various, see how you can, by changing the lines of the facial structure, as you can see, it can almost morph into human. And when you see it, look at the baby chimp, it looks frighteningly close to that of a human being. And so this is the varieties of anthropology. And these are the cladograms, the so-called molecular phylogeny, and as opposed to visual phylogeny. And you can see how the chimps and humans are more closely related, as opposed to the pre-molecular, where they were thought that the gorillas and chimps were closely related, and the orangutans were on a separate limb, and humans were way out there, But it just turns out that the molecular analysis has proven this. And this is based on the more recent evidence of DNA, RNA, and as well as comparing the genomic sequences. Okay, and this is the concept of neo-Darwinism, nothing to do with the movie Matrix, which is in the theaters right now, and that's based on the various changes of mutations, various genes that they are recombined and they're selected out and isolated. So these are the various principles that have formed a new concept. It has come a long way from Darwin himself. Would be pleasantly surprised. Would be interesting to see his reaction. And just to revise your basic biology, as human beings, we are all made of multiple varieties of cells, about 300, 400 different types, starting from a nerve cell, which can be extremely long, starting from brain all the way to the toes, or liver cells, which have a selective purpose to your muscle cells, which responds with changing size and shape, to your red blood cells that does not even have the nucleus, which is an exception to the rule as it is common in biology. While most of the cells have nucleus where the DNA copies are maintained inside, very nicely inside your 23 chromosomes. And these are the various chromosomes that look like a cross-shaped, and these are the telomeres, which have to do a lot with ages. The parts are called chromatids, and the DNA is wrapped around in a tightly wound around varieties of proteins called histones. And those proteins, and it's three or four, actually, to look at very complex three-dimensional structure and folded protein. And if you unwind that, you see the structure, which is the so-called nucleotides, which are the various chemicals that are formed, which is your genetic code, which is about 6 billion letters, half coming from your mom and half coming from your dad, which are in a pair of A, C, G, T. Okay, and they match with each other type of thing. And G combines with C, A combines with T. And even though the DNA, if it's unwrapped, is as long as 6 meters, it's tightly wrapped inside a nucleus that is extremely small, as to a few nanometers diameter. Okay, so, and now some of you may recognize this name, Linus Pauling, a man well known for discovering various vitamins and being putting chemistry on a solid ground, and worked at Caltech, and later on received 2 Nobel Prizes. One was for protein chemistry, the second one was for protesting his nuclear weapons. And he, along with his postdoc student named Emil Zuckerkandl, okay, worked on hemoglobin molecule and found that the hemoglobin molecule had 4 separate rings, and that gives, and based on comparing the various structures of hemoglobin, going back to chimps and other animals, they were able to establish something called the molecular clock. They started working on the crab proteins in France, later on worked on hemoglobin in humans and chimps. And based on the changes in the amino acid sequences that took place, they were able to devise a concept called the molecular clock, which is still been valid and used extensively in mapping the distances on evolutionary scale. Later on, now, it just goes to show you that not all Nobel laureates can be perfect. Even the Nobel laureates cannot be perfect. So he was working furiously on identifying the structure of DNA and made some basic physical chemistry mistake that was spotted by these 2 clever chaps named Watson and Crick. And they were helped tremendously based on the X-ray by Rosalind Franklin, and they were able to come up with the structure of DNA in 1953. And so, but still underlying, Francis Crick realized that protein structure was the important central problem. And then after the discovery of DNA, a race started for deciphering the genetic code of about the 20 essential amino acids that are necessary for survival of human being. And so this was the concept of molecular clock, as to see the distance between the hemoglobin. So it was a linear relationship with the last common ancestor, which was estimated based on fossil records. So this is the start of the so-called molecular evolution. And this is just the basic genetics concept where we talked about that the DNA is kept safe inside a cell, and then it is copied or transcribed into an RNA. And RNA is gradually the messenger RNA brings each and every amino acid in a code of triplets and goes on to translate, put together. The messenger RNA forms something called the proteins, and the proteins are extracted from the DNA and was expelled out as they're being made. And at the same time, they are destroyed in different various parts of the body. Okay, so these are the various ways of the DNA signaling. And the Human Genome Project was started in 1990. It took about 13 years and several $1 billion with the competition of the government, engine, and private industry. And when it was finally decided that the actual amount of DNA that coded for protein was relatively disappointingly small, only 2%. So much so the scientists were frustrated and labeled the rest of the DNA as junk. It turns out that it is not junk. In fact, this is an extremely complex orchestra and mechanism of feedback and control loops at a great distance from the original coding sequences, trying to respond to various environmental and signals that takes place. So the genes are turned on and off. So the genome evolution goes on even after you're born, and depending upon the exposure to ultraviolet light, food, nutrition, stress, all sorts of factors influence. So again, it's a nice choreography of nature as well as nurture that goes on. Okay, so this is so the genes are turned on and off. So that's how the liver cell only produces liver protein and does not go around making red blood cells, and vice versa. Your bone marrow stem cells only produce red blood cells and don't produce insulin. So that's conducted in a various precisely controlled forms. And if we see the intracellular signaling pathways, they're just much more complicated than the underground network of the internet. And so the varieties of genes are sensed and non-sensed. And so some of the sense genes are interspersed with non-sense elements, okay? And at the same time, introns, the exons are the ones that code the proteins. They're interrupted by a segment of long sequences, which don't seem to do anything apparently, and they're called introns. And so the varieties of, now when we say, this was an interesting statement that I came across, that the junk DNA is not actually junk, garbage, but it comes in useful in a varieties of ways. Okay, so now, and when people started comparing genomic sequences, a lot of the sequences that you and I have is similar, goes back even to the slowest organism, such as a fruit fly. And so there is, and the reason why it's not altered, because it's extremely important. Without that, the animal would die. So the changes that are seen are the ones that are that do not harm survival. So these are varieties of regulatory elements that specify so. And the structure is about a three-dimensional structure. It is folded on itself. And so, and so this again, just as you may have recalled this from a long time ago, that God does not make junk, or at least the DNA does not make all junk. It's all useful. Now, this may shock you or surprise you that the viruses, that part of a huge amount of your DNA is actually all viruses that got inserted somehow spontaneously inside your own genome and made home. And this is not only just in human beings, but even in chimpanzees. So there are as many as 200 plus elements of the sequences are shockingly similar to that of a virus that are in your gene. Okay, and out of this, okay, and so you can see there are only 7 sequences that are different between the from the retroviruses, and even in the chromosome, there is the chimp has 1 less chromosome because part of the chromosome broke and it got attached to the second chromosome. So the second chromosome in human being is a lot larger than that of a chimp. Okay. Now, again, some of the genes that are have stopped working in human beings. One of the very easy example is something called the pseudogene, which they could manufacture vitamin C while we have lost the ability, and because their diet was primarily fruit. Okay, and so there are varieties of changes that have taken place. Why that they occurred, it's not known well as to, but somewhere along the line the gene is present, but it has lost its function. Okay, and the other, the person who did the comparative work earlier was a woman who is well known from her work in other fields, that she discovered the BRCA gene, which was for breast cancer. It's an interesting story by itself as to how she was in a race with some well-funded researchers from the University of Utah, because they had access to a large amount of data from the family trees of Mormon families. And she discovered the BRCA gene from a patient named Annie Parker, who had a family history of cancer, and it was made into a movie starring Helen Hunt, decoding Annie Parker. And this is a woman who discovered the BRCA gene named Marie Claire King. She did her work initially in California and earlier in Michigan as well. And she worked with a scientist, geneticist named A.C. Wilson, who was out collecting fossils and evidence. At the same time, she did the protein comparison analysis between chimps and humans, and was shocked to find that 12 out of 12 protein sequences were similar. And we talked a little bit about the molecular clock. And later on, with the similarities, a major project was undertaken, something called the Human Genome Project. I'll pause for a few minutes and then we'll continue in a few minutes after I get some water. Okay, so we're talking about the so-called Human Genome Project as to see as even though earlier I showed you there was only 1.85% difference, but that's not exactly true, because even though the DNA may be similar, it's kind of rearranged like a deck of cards, is reshuffled, copied, deleted when compared to. So it's like, and only when it comes to the important stuff, that's the proteins, that only about 1/3 of the proteins are 100% identical. So in reality, even though the sequences may be appear similar, the actual work that they do is, there's a substantial difference. So, and when we look at 5% of DNA is rearranged, and when we talk about several billion letters, that's still 150 billion letters. So, and it only takes sometimes one letter change to make a dramatic difference in the outcome. It is well apparent in the classic example of sickle cell disease that the one amino acid sequence can cause serious disease in thousands of people. Okay, so this is, and then again, there are certain things that are which have more, we have more copies. For example, the gene that is digest the starch in our mouth called amylase, and the chimps have only 0 to 1 copy, while humans have several copies, about 2, or some have more copies. And again, depending upon our diet, the copies that are there of this particular gene and corresponding enzyme in our mouth is different. Vegetarians will have more copies. Similarly, lactose gene, which is turned off after a few years in baby chimps as well as humans, but we have reacquired that ability. Okay, so so we are similar, but still quite different, and we'll show as to where the big differences are. How this sequence comparison, because this is how the clade tree is formed. Gorillas out on the other limb, much further away. Bonobos, chimps, and humans similar. This is this 6 million years difference that took place, and so this is the most of the sequences. You can see how I'll try to zoom in on here. The humans, chimp, and mouse, several letters are very similar, but when it comes down to this thing says the mouse does not have sequences which humans and chimp do. And even here, we can see there are similarities that take place between. So this is, gives us the idea of the rate of substitution that takes place. And then further, more detailed comparison shows the differences between the two. So this is a so-called classic example as to how the sequences outlined with powerful computers and in public domain appearing browser. Okay, so that's how the substitution rate and the comparison is carried out in a very meticulous manner. Okay, so as I showed earlier, that our humans are basically baby chimp, and there are certain regions, and then the classical, the biologically term is called pedomorphism that has been given. Okay, and so this is an example that is given. Okay, and they express different levels of protein that control facial developments, jaw, nose length, skin pigmentation, etc. that take place. So there are various insertion deletions that take place. There are something called copy number variations that take place. It means we have more copies, So, and we talked about how the mutation gives us an idea about the dates of separation. Okay, I think I'm going to stop this share for a second and see if I can find another video that I can share with you guys. Okay. Let me see if I can find it on my computer. Okay, let's see.
Welcome back. So when we line up genomes, we're looking at what has changed and what has been preserved in the chimp and human genomes since they diverged from a common ancestor, perhaps our
Sorry, I
friend
think you may
Sahelanthropus
not be
here,
getting the
7 million
sound for
years
whatever
ago.
reasons.
Take any stretch of human DNA and the stretch of chimp DNA that corresponds to it, they'll represent the same piece of ancestral DNA passed down in 2 separate lineages over the course of 7 million years. Now if we take a sequence, and this is million intended to be gibberish up here, from a chimp and a human, and you put it through a computer, you're going to find hidden messages. This is hidden. This is hidden. Computers are good at finding this kind of thing. Stuff that's better preserved than it should have been just by chance, and stuff that is different. We call the ability of computers to extract useful information from genomes bioinformatics. And of course, given big data, computers are crucial. They enable us to ask questions and get answers in ways we never have before. Applying bioinformatics to the human and chimp sequence, we find about 98.5% of it is conserved. And it's 1.5% that's interesting because it identifies a set of mutations that accumulated in the DNA of the chimp lineage, and some of these were made a chimp, and another set of mutations that accumulated in our lineage, making us humans. So how do we differ from chimps? Well, about 1 in 100 base pairs is different. That's compared to an average of 1 in 1,000 between you and me. And given that the genome is 3 billion base pairs long, that means there may be 30 million human-specific letters of code that are not shared by the chimp. In addition, there are about 5 million indels, which I remember means insertions and deletions in the genome. And they're mostly in regions that don't encode proteins. And that means that not many of those 5 million indels will contain genes. So there's not going to be many copy number variations in us and chimps. Now, bear in mind that there are copy number variations between us. So I may have more or less genes than you. But if we look at those genes where modern humans all seem to have the same number, then about 30 genes are replicated in us since we split from chimps. That's not many, so we have a very similar number of genes as a chimp. But interestingly, a lot of the extra genes we've got are involved in brain development. The best studied of these is a gene called SRGAP2. Genes have names like that, don't worry about it. Anyway, this gene duplicated 3 to 4 million years ago when we first began using tools, and again 2.5 million years ago when our genus Homo separated from the Australopithecines, who had brain sizes not much larger than that of modern apes. So what that means is that we now have 3 copies of the gene, whereas chimps only have 1. Studies of mice have shown that having these extra copies of SRGAP2 would have changed our ancestors' neurons so they developed complicated shapes that made them capable of exchanging information with a larger number of neighbouring cells. So SRGAP2 teaches us that evolution wasn't just about giving us bigger brains, it also changed the way our brain cells interacted. Now, something else interesting about these duplications is that they would have changed brain development immediately and dramatically. So human ancestors with 2, 3, or even more copies of the gene could have coexisted at one point, which is kind of fun to think about. Well, a jury is still out on the impact of most of the other indels, which is not surprising as we're still trying to work out the impact of the indels on copy number variants in human variability. So scientists comparing humans and chimps, they've mostly focused on those SNPs. It's been estimated that out of the 30 million SNP differences with a chimp, perhaps 10,000 were changes to protein coding genes that altered our bodies and therefore were subject to selection. And on top of that, there's going to be mutations to regulatory regions of our DNA that turn genes on and off. So the questions are, which of those particular DNA changes actually made us human? And how do we identify the sequences that make us human? Well, to find out which of these spelling differences in DNA sequences are most important, Catherine Pollard at the University of California in San Francisco has developed powerful computer software for scouring the genomes of human chimps and other vertebrates. She's looking for hotspots along the genome where DNA has changed especially rapidly in the lineage that led to humans. Pollard and her team reasoned that DNA sequences that had undergone the most modification since a human-chimp split were the sequences that most likely shaped humankind. So where chimp and monkey and mouse and cat and chicken and fish have exactly the same letter at a certain place for hundreds of millions of years, it suggests that evolution wouldn't let it change because it was needed for an important role in these animals. So the Pollard lab has thus far identified 202 cases where there's a sudden burst of change in a DNA region in the human lineage, and they call these recently changed regions human accelerated regions, or HARVs, H-A-R-S, HARVs. Now what's really interesting was that a lot of the sequences that are really different between us and chimps don't encode proteins. So that means proteins don't have it all their own way in evolution. But then we could have guessed that, right? It comes back to the fact that a lot of the 98% junk matters. It contains regulatory sequences that tell other genes when to turn on and off, as well as a whole lot of DNA having purposes that scientists are only just beginning to understand. The majority of HARVs seem to be involved in gene regulation, because they are sitting in front of genes, or at least near them. So for these HARVs, Pollard and her researchers figure out what gene it's next to, and then they ask, where and when is that gene switched on or off during embryological development? And then, is its expression pattern different in the human than in a mouse, a rat, or a chimp? If it is, then the novel pattern may prove to be relevant to an understanding of how humans are distinctive creatures. Well, HAR1 tops the list of the most divergent sequences between us and chimps, and it's an exciting finding because it seems directly linked with our braininess. So look at this table. It shows the HAR1 sequence in us, in chimps, and in chickens. This region of the genome, as you can see, has changed very little for most of vertebrate evolution because the chimp and the chicken sequences here differ by just 2 letters. Well, the fact that HAR1 has been essentially frozen in time for 300 million years since the chimp and the chicken had a common ancestor indicates that it does something very important. However, the human HAR1 has undergone 18 substitutions. That it underwent abrupt revision in humans suggests that its function was significantly modified in our lineage. Now, the real exciting thing about HAR1 is that it's in a genetic region that is turned on early in neurons that play a key role in developing the neocortex, the wrinkled outermost brain layer. When things go wrong in these neurons, you get a cerebral cortex that lacks folding, a very sad condition called lyssencephaly. Now, it's not known what HAR1 does in those neurons, but it's just active in the right time and place to be instrumental in the formation of a healthy cortex. Thus, potentially, HAR1 could have altered ancestral human brain function. We don't really know much about how the human cortex develops, but the HAR1, the HAR1, provides a new lead and a target for experimentation. So aside from braininess, what other feature is often used to distinguish humans from our fellow apes? We're the apes with the big brains, and we've got the opposable thumbs. So would you like to hazard a guess? What do you think HAR2 does, HAR2 does? Well, it turns out the second most divergent sequence between us and chimps is a regulatory region, an enhancer, that drives gene activity in the wrist and the thumb during fetal development. Well, that's particularly interesting as the ancestral version in other primates doesn't do this. The human HAR2, perhaps therefore contributes to human-specific hand coordination and the dexterity to manufacture complex tools. Now, please note, that doesn't make chimp hands inferior. Chimps don't want a thumb. Chimps swing through the trees. All you need is a little hook for that. A thumb would get in the way of a chimp and its lifestyle of choice. Now, there are several other preliminary stories relevant to the brain. HAR152, for example, is near the gene encoding a protein called neurogenin 2 that's expressed in a region of the hippocampus. We'll talk about the hippocampus in later lectures, but the hippocampus has a central role in learning and memory. Pollard's group believe, though, that once all the experiments have been done, it's going to turn out that more than half of the halves probably upregulate genes that do things in our brains. Thus, even though halves make up a minute portion of the genome, changes in these regions could profoundly alter the human brain by influencing the activity of whole networks of genes. Have a look at this model. You have a master regulatory sequence that perhaps encodes a regulatory protein which switches on a battery of other genes. Some of these in turn switch on other genes, so you get a cascade of changes just resulting from one sequence changing. Perhaps this downstream gene here produces an enzyme that makes a hormone. Mutate the gene, that particular downstream gene, I'll produce slightly more or less hormone, and I'll be a little different than the other kids on the block. But if I mutate the master regulator downstream here, upstream here, beg your pardon, I could affect hundreds of genes. It'll have a radical impact. It's possible I won't even get to be born. But if I am born, I'll certainly be different, and natural and sexual selection will say if it works. It may be then that the way to evolve a human from a chimp-human ancestor without adding much genetic innovation is to change sites where changes make an important difference in an organism's functioning. By changing regulatory sequences that tell genes when and where to turn on and off, you can redeploy the existing squad of genes in new and interesting ways, and the results can be dramatic. Like changing a sports manager could have a much greater impact on a team's performance than just switching out individual players.
Okay, so that was interesting. Let's start a different share. Okay, here we're so this is, I'll set up my slideshow again, and from the current slide. Okay, so we already talked about this one, human accelerated regions. Nietzsche said that humans, all two humans, and this is, we already saw this slide in their lecture type thing. So, okay, all right. And now this is something interesting that was not talked by the lecturer here. And this I just came across that nowadays, people can take embryonic cells, turn them into pluripotent stem cells which are basic fundamental neurons. And these are the comparisons of human brain organoid. And by ethic law agreement, they're only allowed to grow them up to 14 days. And you can see that even at that early an age, that the human even at okay, yeah, I'm sorry. I think I goofed up or something. So, okay, the video thumbnails are minimalized. Okay, so this was the experiment that we were talking about earlier. At 5 weeks old, the human organoid is far bigger than the gorilla that we saw earlier. Okay, in the chimpanzee, the gorilla is at top right. So this is a scary example as to how changes can take place. And this is another way of showing you how that takes place between the this was a different paper on the Hars-Noel gene as to the human accelerated regions are relatively small letters, but you can see the dramatic difference that makes. It's again, as the professor talked about from Maryland, as to it takes place in the outside the genes, and this again, so dramatic developmental changes take place. So between various, you can see that as the limbs are enhanced, HARS1 and HARS2 that he talked about earlier. You can see at the embryonic level, the changes take place. This was a paper which is fairly old by science, Dr. Pollard papers of HARS1 as early as 2006. And this is changes in the limb development that takes place between chimp and human accelerated regions. Okay, and sorry, go back. Okay, so these are the various proteins that the so-called Notch1 protein that takes place, and this is named after fruit fly. Okay, an area in the fruit fly where they had a notch, and that's modified in humans, as well as the SR gap protein that takes place in the brain, as we talked about, which has to do something with the dendrite growth. Then the other gene, which is called zinc protein, that has to do with cognition, whereas glutamate, which is an important metabolite in your brain, okay, and sialic acid, that has to do with your digestion, and MYH16, that has to do with jaw muscle formation. And so these are the varieties of changes that have been discovered so far, and I'm sure there are many more. Okay, and this is again shows how the turning on to the gene changes the expression and the formation of the brain size different. Okay, the neurons differentiate when the gene is turned on, and the growth is goes faster than differentiation. So that's why we're able to grow a larger brain. Okay, and again, this is shows the example of the brain size that we talked about earlier. As starting out from Sahelanthropus to RD to the other branches that separated out, and then Homo erectus, and then Neanderthal. And so this is you can see that the tree is rather diverse and marches out in different directions. Now, so this brings us to the point of eventually it's been well established that all of the human migration took place out of Africa in two different streams that took place. One went through the Middle East, through the present day Sinai Desert area that the migration took place. And another took place in present day Saudi Arabia area. And so this was supposed to have taken place around 300,000 years ago. And then in another 100,000 years, they were all the way out in Europe. And then by 70,000 years, the human habitation evidence has been found as far as Australia. So we got around rather fast, one can say, in terms of, because obviously, so the dispersed, it was not, and obviously there were other species already there, with so it was not a colonization, it was not. So it just started, the expedition that out of Africa. So that theory has been well proven, and so the migration took place twice. One, the skulls have been isolated in Israel, which are as old as 130,000 years old, and there's one that is even earlier. Okay, in 2002, and based on uranium dating in Arabia, fossils have been found place, and there was a passage through the Red Sea area, which is called Baba el Mandeb. That's the other migration that seems to have taken place. Okay, so this were some of the sites where human fossils have been found, and this was the original fossils that were discovered in South Africa and Rhodesia. This is the so-called cradle of human civilization, where Dr. Lee came. And the most recent one is in a place called Jebel Erhoud in Morocco. And these are the Sinai excavation sites. And this is another route of human migration that took place, as you can see, through the narrow isthmus that took place. And these are the various excavations that have been done in Israel. These are very apparent as well as in Greece. So various places where the fossils have been found. The migration out of Africa still continues, as you can recognize by this man who is trying to change the world in his own way. And this is just another way of showing prehistory takes place. As you can see, that's time scale and the various different ways of forming. So for 4 million years, the Australopithecus, Afarensis, Lucy, okay, and then the hominids started making tools. The first members of the Homo clade with a larger brain 4.2 million years ago, okay, and then 150,000 years ago, hand axes were used, okay, and then there was the rise of Neanderthals in Europe, and then in the last 100,000 years, the Homo sapiens came to being and took over the world literally. Now let's see if I can.
The story of humanity's evolution began about 7 million years ago, when the human lineage broke away away from that of chimpanzees. Over time, an ensemble cast of over 20 early human species, or hominins, came to the fore. Most became extinct, while others might have been ancestors to today's humans. Each species exhibited varying degrees of human-like physical and behavioral traits, such as large brains, small teeth, bipedality, and tool use. These hominins fell into three major groups: early hominins, Australopithecines, and Homo genus. Humanity's earliest relatives lived between 7 and 4.4 million years ago in Africa. Having most recently shared a common ancestor with chimpanzees, they had many ape-like traits, such as a small cranial capacity. However, fossils show that some ancient hominins were also beginning to show human-like characteristics, such as small canines that were likely used more for eating and not for hunting or fighting. The next phase of hominin evolution involved primates called Australopithecines. They lived between 4.4 and 1.4 million years ago across the African continent. Like their ancient brethren, Australopithecines had some ape-like traits. However, changes in the skull, spine, and legs indicate a notable shift toward a very human-like trait: consistent bipedal locomotion. The third and current phase of human evolution involves members of the genus Homo. The earliest Homo species likely date to more than 2 million years ago, making them a contemporary of some Australopithecines. But unlike earlier hominins, who exhibited a mosaic of ape and human-like traits, Homo species were becoming distinctly more human. Their cranial capacity was growing larger than any other hominins. They developed sophisticated stone tool technology, and they became the first to control fire. These physical and behavioral adaptations, along with advancements in technology, allowed some Homo species to be the first to migrate out of Africa.
Okay, so you get the idea. So this was a nice clip from, I think it was from National Geographic. Okay, and this is not just based on fossil evidence, but it's also been proven with genetics, type of thing. So the genetic variation that has been shown is there's a much more genetic diversity in Africa as opposed to, because that shows that they were the older genetic tree. And so that means that the amount of diversity is people living in the same area. So it's the same thing with Europe and Central Asia. So the last to be habited was New Zealand area. Okay, so there are various mathematical calculations have been done on a very sophisticated, and based on that, it showed that the earlier number of people that migrated were relatively small, at the most 10,000 or so, and they could have been nearly wiped out due to climate changes and other changes. So it was a lucky incidence. Okay, and this has been proven with various bones, stones, and genes. So this were the. So we already went through that. Now we'll focus on Neanderthals and Denisovans, which are the two prominent species. And the Neanderthal discovery was done rather accidentally in 1856 in a cave near Neander, which is near Düsseldorf in present-day Germany. And these were our nearest relatives that inhabited a large area. Originally, the discovery was confined to the glacial areas of Europe, but later on, it has become apparent that their range extended much further into Central Asia. And so, in the fossils, as to why they disappeared, there are various theories as to why they disappeared, but nothing has been established. Some of the characteristics that they had were very similar to us. They were social creatures, and they were hunter-gatherers. They involved using of tools, art, cave art, culture, and so and then certain based on the modern DNA analysis that was carried out by the Max Planck Institute in Germany by a guy named Swan Pabo, who's I think from Finland, and they were able to isolate ancient DNA, and there was a remarkable discovery, and that showed that some of the DNA is still preserved in modern day humans. And most of the Europeans carry anywhere from 2 to 3% of Neanderthal gene. And some of the genes have been beneficial in our immunity development, but other genes have been sort of have had a mixed bag of the inheritance. For example, when you may be surprised that the gene for waking up early is inherited, it goes back to the Neanderthal days. And even the gene for red hair traces back to Neanderthal days. Okay. And subsequently, most of the European gene has been replaced by the invaders from the Caucasus that took place. And earlier it was thought that they were much more closely related to the farming migration that took place from the Middle East, but that we'll see later on as to how that turns out not to be the case. Okay, and similarly, from a tiny pinky finger bone that was isolated in a small cave in Russia in 2009, a totally new human species of Denisovans has been identified. Okay, and this is, the artist rendition of Neanderthals that had rather prominent brow ridges. Their eye sockets were bigger, and as to why they had the prominent brow ridges, that nobody knows. And this is based on artist rendition, and they love to decorate their faces with feathers, and they lived in caves in family groups. Okay, and they used animal hides. Okay, and they made jewelry out of shells, and the cave art has been discovered. Okay, and there is, and they also extended further out into Central Asia and were contemporary of Denisovans, and evidence of inbreeding has been found especially in a skeleton that was discovered in a cave in Romania. It showed that it had a genetic mix-up of both Neanderthal as well as Denisovans. Okay, so, and this is the Neanderthal range in more details as we talked about. As you can see, it extends all the way from Spain to Malta to France to Britain and all the way to Central Asia. So they got around for sure. Okay. All right. So they're relatively, one can say that they're cousins and not nearly ancestors. And even then, further skeletons have been identified in the 20th century. Complete entire skeletons have been identified in southern France. Okay, and also a way based on some cave drawings, there is clear-cut evidence of damage to the bones of ritual cannibalism. Okay, so that may be a surprise now. They were also being based on the analysis of the teeth. It was apparent that they not only were hunter-gatherers, but they also had used medicinal plants. Okay, and so this is, and as those of you who have good our movie buffs may remember, there was a movie and based on a series of books by a lady named Jean Auel, which was called Clan of the Cave Bear, and that was remarkably spot on, so to speak, because it was a kind of a historical fiction and based on some of the archaeological and geological evidence about the cohabitation of both modern humans and Neanderthals. So it's a really nice, well-written book, and she was way ahead of her time. Okay, and the earlier concept in the early 19th century was that the Neanderthals were all brutish and they were much more close to apes rather than modern humans. But that has turned out that we were totally wrong in assuming that. So there was nothing brutish about them. This is some of the shell necklace that they had formed. This is the tooth necklace. There was cave art that is as far back as 65,000 years old in Gibraltar, modern day Gibraltar. And this is so you can see that there was interbreeding between the modern humans, Denisovians, and while the Europeans do not have too much of Denisovian genes, their Denisovian genetic sequences have been identified all the way in South Asia and Papua New Guinea. So they got around. There was a substantial interbreeding that took place. So did the archaic gene flow matter? And there was a lot of interbreeding that took place. And so much so to remember this commercial. And so, and some of the stuff that we have inherited from, I already talked about what we might have inherited from Neanderthals, but from the Denisovians, especially the Tibetan population, may have inherited a gene that gives them adaptation for surviving in cold air. And so that it may have occurred as recently as a few thousand years ago. So this is, and some of the immune genes are also well preserved from the Denisovians, and they've been traced down in modern day Indonesians. Okay, so this is, it just goes to show that there was a lot of, so, and this is a slide from Dan Lieberman. He talks about the running abilities of human beings. Okay, but I think that is well apparent. We talked about as to how, you know, a lack of, our long bones, as well as absence of hair, as well as sweating ability, gives us the ability to outrun a dog or even a horse, especially if you run in team for long distances. Okay. And this is the range of the Denisovians, as we can see that this is a part of Russia, that the small area that can be magnified, and you can see that the Denisovian caves are at on the bank of this particular small river that has been isolated. And not only just the Peki, but as recently as May of 2019, a jawbone has been isolated. So the separate existence of the species have been well documented with genetic evidence. And now next time we'll talk about this new species that was discovered in various parts of Africa, and we'll continue next time. So I'll stop the recording right here, and I'll stop my screen share. Okay, and I'll try to